Welcome Bio 2600 students. Now let's dive into sensory physiology, where we will discuss first off general senses, and then we'll move on to the special senses. First, let us try to figure out what are sensory receptors. So receptors are modified parts of the peripheral ends of the neurons, sensory neurons, so your afferent neurons. The peripheral ends are switched up a little bit. They are modified to pick up changes in their environment. The changes or these stimuli could either be external stimuli, for example, pain, touch, pressure. Those are from the outside of the body. Or they could be stimuli from the inside of the body. So internal stimuli like changes in oxygen levels, blood pressure changes, changes in pH levels. Okay, so again, we have external and internal stimuli. And because and when these receptors pick up changes, that will lead to a local graded potential. Remember, these local graded potentials are, they do have the ability, if they do reach the threshold level, to become a full action potential. And that's when we will actually experience that sense. Now we have 5 major classes of sensory receptors. So there's so many different stimuli that we can perceive, and that's why we have lots of receptors to pick up, the wide variety of stimuli. So we have mechanoreceptors. These receptors pick up changes or mechanical changes. For example, touch, pressure, vibration, all of these are picked up by mechanoreceptors. Thermoreceptors, well, as the name indicates, these pick up changes in temperature. Photoreceptors, these are only found in the eye because they pick up visual, they pick up light. Chemoreceptors pick up changes in chemistry, so those are found, for example, in the taste buds. They are also found in the nasal cavity. Nociceptors, these receptors pick up pain. Now for the kinds of receptors, like where are we going to find these receptors? There are 2 different ways. Either the receptor is part of the sensory neuron, is actually right on top of the cell membrane of the sensory or the afferent neuron. And you can see here these purple receptors that will be stimulated in the presence of the stimuli, the sensory stimuli. On the other hand, we have what are called receptor cells. Receptor cells are modified neurons or specialized neurons that pick up changes in the environment. And again, you could see here these receptors found on the receptor membrane. So there's a little difference because we keep on using the term receptor. I want to make sure that you are aware that there are 2 different things, distinct things that we name receptors. So remember receptors, we talked about nuclear receptors and cytoplasmic receptors that would attach to a chemical ligand, a hormone, for instance, and lead to protein synthesis and so on. So that is one kind of receptor. The receptor we're talking about here, whether it's, these or this receptor cell, is a cell that receives stimuli, receives the change in the environment. So again, make sure that we're not confusing these 2 different kinds of receptors. In the presence of a receptor cell, that receptor cell has receptors on its own membrane that pick up the changes in the stimuli out here. And then through these neurotransmitters that are stored in the vesicles, these neurotransmitters would be released, would stimulate these receptors in order to take that sensory impulse into the CNS. Now, adaptation is a process where you keep on stimulating these receptors so much that they stop responding. So adaptation is the decrease in receptor sensitivity, which results in the decrease of the action potential frequency in an afferent neuron, despite the constant presence of a stimulus. So I'll give you an example. Let's say you have, this nagging kid that it keeps on talking all day long, or at one point. So that is definitely activating your maybe visual receptors, your auditory receptors, and so on. But because it is so repetitive, there is a point where the kid is still talking, okay, but you are no longer aware that he or she is there. That is known as adaptation. So there are different reasons for adaptation. One of them would be decrease in the receptor sensitivity. Because of the frequent stimulation of the receptors, these receptors have now adapted to it and are no longer really picking it up. Okay, so it is being picked up, but it doesn't really reach the brain where the person is experiencing these stimuli. So an adaptation is the decrease in receptor sensitivity that results in the decrease of the action potentials reaching the brain, although the stimulus is still there. Okay, so we have receptors that slowly adapt and other receptors that rapidly adapt. The receptors that slowly adapt are known as tonic receptors, and these are the receptors where you kind of do not want them to adapt. So for example, pain receptors. Pain receptors are very slow to adapt, or maybe they don't adapt at all. And pain is a protective mechanism. As long as there's pain, we want to go and figure out what is the problem. So you don't want that pain to be removed because, again, it's a red flag that there's a problem and we would seek medical advice for it. Other slowly adapting receptors would be the receptors that are found in the muscles and tendons, and it sends information to the cerebellum and different parts of the brain to maintain equilibrium. So these are receptors, again, that you do not want adaptation to occur. Other receptors are rapidly adapting, and those are the ones related to smell, taste, and touch. Okay, so you put on your socks in the morning or your shirt in the morning, 2 seconds later, you no longer feel that it's there. When you smell something or taste something, you could really experience it in the first 1 or 2 seconds, and then after that, we lose that experience. We have to take another bite and so on. So these are rapidly adapting receptors. They're known as phasic receptors. They have what are known as on and off response. So when you're first exposed to that smell, you experience it. And then when the smell disappears, you experience it again. So here you can see we're plotting here. This is when the stimulus starts, and this is when the stimulus ends. Up here, these two lines up here show you the rapidly adapting receptors. So for example, again, the taste and smell receptors at the very beginning of eating that bite, you could experience the taste. So that's when the stimulus starts or the stimulus is on. When the stimulus is off, so when you swallow that bite, that is again when you experience the taste one more time. But really when you're chewing, there's really not much stimulation of the taste receptors. In the case of slowly adapting receptors, so for example, pain, this is where the pain starts. So in the on, you can see that the frequency of the action potentials are high, so the intensity of pain is high. As the day goes on or the days go on, you're still experiencing the pain, but it's at a much less level, and that is interpreted in less frequency action potentials. Now, stimulus type or stimulus modality means that we have different kinds of stimuli. So we have stimuli that could be heat, cold, pressure, sound, light. These are just examples. There's just so many more. Each modality would have a sub-modality. So for example, thermoreceptors could either pick up cold. We have some receptors for cold and other receptors for hot. So these would be sub-modalities of temperature. And we have a specialized receptor for each modality. Now for primary sensory coding. Primary sensory coding means it is that the stimuli come in different ways. So we have mechanical stimuli, there's chemical stimuli, photostimuli. How is it that all of these different stimuli are converted into an action potential, and that's the only way our brain can actually understand them? So coding is the conversion of these stimulus energies into a signal that conveys the relevant sensory information to the CNS. So how is it that vision or light that is picked up by the eye, how can we convert that into an action potential? That is known as primary sensory coding. Okay, so all the different stimuli that we experience, all of them, some way or another, have to be coded into an action potential, since the brain and the spinal cord only understand action potential. Not only do we experience the senses, but we also are able to analyze it. So what is the intensity? How strong is it? What is the location? And, I'm going to just use this image for the next couple of slides, where the receptors, for example, on the back are able to, or unable actually, to identify these 2 points. Okay, that is known as tactile discrimination, where on the back, you really do not need to identify 2 different points on your back unless they're far apart from each other. While the tips of the fingers, we do need that for fine touch, and the tactile discrimination is a lot smaller, where you are able to identify 2 different points. And on the tongue, you see it is even smaller than that. Okay, so that again, comparing the need of each of these different parts of the body, we are able to identify the exact location, okay, and the intensity of these different stimuli. Now, why is that? How is that? How does the body code for intensity, and how does it code for location? So let's take a look at this sensory neuron. You can see here that is the cell body, and we're talking about general senses. So this is a unipolar neuron. You can see the central process going to the CNS, and the peripheral process will be going to, in this case, will be going to the skin. At the end of the axon, you see it branching off, and all of these are the axon terminals with the little receptors at the tips. And all of that would be considered the receptive field. So if you stimulate any part of that skin, that would lead to an action potential in this particular neuron. Now again, how do we code for the intensity and location? And that's what we will be talking about in the next slides. So for intensity, we'll do this experiment right here, where we'll get a glass probe, and this is one sensory neuron, and that's its receptive field. We'll apply a little bit of pressure, not a lot. So you see here that none of the receptors are activated, and that translates into no action potentials. Now, if you push a little bit harder, so you apply a little bit more pressure, that is going to activate some receptors, and that will start the creation of some action potentials. And as you gradually push harder and harder onto the skin, you are gradually increasing the number of receptors that are being activated, and that translates to higher frequency of action potentials. So the body or the brain is able to translate the intensity of the stimulus through the frequency of the action potentials. So higher stimuli or stronger stimuli are going to recruit more receptors, and that is interpreted into a higher frequency of action potentials. Now here you can see that little piece of skin. We have in this case 3 neurons. We see here this is the receptor, the field, the receptive field of neuron A, and there is a little bit of overlap with the field of neuron B, which overlaps with that of neuron C. So how is it that, again, we are able to localize that the stimulus is in this spot and not related to A or related to C? Okay. So what happens is that if you apply the stimulus right here, right in the middle of the sensory field or the receptive field of neuron B, that will lead to more activation of neuron B. It'll still activate neurons A and C, but not as much. So you see that the frequency of the action potential is definitely less when it comes to neurons A and C, but a lot higher at neuron B. So that will be interpreted in the brain that the location of the stimulus is related to this sensory field or this receptive field and not A or C, I'm sorry. But again, we do have a little bit of an overlap, so it's not very accurate, but the body can overcome that, and I'll show you how. And that's what we were, mentioned a while ago, the fact of 2-point discrimination. And that means the ability to discriminate between 2 points when they're stimulated at the same time. So in areas of the body where you really do not need that 2-point discrimination, the sensory neurons innervate larger parts of the skin. So whether you can see here there's a huge sensory or huge receptor field, whether these 2 points are stimulating this part or that part up here, you're still stimulating the same sensory neuron. So it really doesn't matter. The brain isn't going to be able to pinpoint exactly where it's happening. It will say that this is just 1 point, not 2. While in areas where there is bigger discrimination, so for example in the tips of the finger, the tongue, and in the lips, you can see here that the fields are much smaller. We have more sensory neurons. And these 2 points are stimulating 2 different sensory neurons. So you see here, this one is stimulating the orange, while the one on the bottom is stimulating the green. And that will be interpreted in the brain into 2 points. So this individual will feel that there are 2 points, while here that person will not be able to discriminate between the 2 points. That person will think that they're only being pricked in 1 point. Because again, both of them are stimulating the exact same sensory neuron. You can see that over here as well, where this is one sensory neuron. That's the skin again. Whether you're stimulating A or point B, both of them will stimulate the same sensory neuron. So you would have that very low 2-point, the low ability to discriminate between the 2 points. And this is what happens in the case of the back. But if you're in an area where you have lots of sensory neurons, the brain will be able to interpret these 2 different stimuli as 2. Okay, and that again would give you that ability to discriminate between these 2 points. So this individual will say, yes, there are 2 points being stimulated. In this example, they would say that there's only 1 point stimulated because both of them are stimulating the same sensory neuron. Now, we talked about overlap of these receptor areas. Now, how is it that even though there's this overlap that we talked about, where did it go? Overlap of these receptive fields, how is it that the brain is still able to pinpoint exactly where pain or a stimulus is coming from? That is due to what is known as, there are several reasons. One of them is known as lateral inhibition. So let's take a look at what is going on here. You have 3 different sensory neurons. We'll name them A, B, and C. You can see that there is overlap in their receptive fields. But this is the area that is being stimulated. So the area stimulated is basically right in the middle of area B. But the receptive fields from A and C are still being stimulated. So remember we said that neuron B would be firing up a higher frequency of action potentials, while A and C will be sending out action potentials, but at a lower frequency. So that's one thing that the nerves do. Another thing is lateral inhibition, where because the action potentials from neuron B is the most, it would send out these inner neurons to inhibit the action potentials coming from both A and C. As you can see here, these orange interneurons are exclusively within the CNS. Remember, interneurons do not leave the CNS. And again, these would be inhibitory from the neuron that is being mainly stimulated, so the neuron with the highest action potential frequency, inhibiting A and C, and those are the neurons with the lowest action potential frequency. So lateral inhibition helps us localize a stimulus, okay, because now that nerve B was inhibiting the impulses coming from A and C. Now the brain will interpret that as the painful stimulus coming from the skin area supplied by B. In lateral inhibition, information from the sensory neurons with the receptors at the edge of the stimuli, although they are being stimulated, so A and C are still being stimulated, but they are inhibited through that inner neuron, causing that lateral inhibition. So less information or a lower frequency action potential will be sent from A and C to the brain. Now that lateral inhibition really enhances the contrast between the frequencies of action potential. So again, the brain is able to localize the site of the stimulation as being part of the skin innervated or innervated by neuron B. So that lateral inhibition pretty much removes the information from the peripheral regions. And we really need that where localization is really needed. So for example, in vision, okay, when you're looking at an object, you really want to be able, your eyes need to be focusing on that object. So there's lateral inhibition of things coming from the other objects that you're not focusing on. And touch. Okay, so both vision and touch have significant lateral inhibition. Other things like pain and temperature do not. So if you've experienced pain or a burn, the area that is painful is pretty much, a bigger area. It's hard to pinpoint exactly where the pain is coming from. You've got patients that would come in and say, like, this part of the abdomen is hurting, and we ask them to pinpoint where it is. It is sometimes hard to do that, okay? And that's because pain and temperature do not have or don't exhibit significant lateral inhibition, while vision and touch do. Lateral inhibition so that lateral inhibition was from the sensory neuron, okay? There's other inhibition that comes from higher centers in the brain. So let's take a look at what we're looking at here in this image. That is your sensory neuron. Okay, so this right here is the peripheral process going to the skin with your receptors at the end. This is the central process going to the brain and spinal cord. This is the ascending neuron. That again will be ascending in the spinal cord to get to the higher brain centers in the brain. Now we have these descending pathways. Descending means motor pathways. So they're both descending in the spinal cord from higher brain centers down. These red ones are interneurons. You see them connecting the motor to the sensory, and again, they are exclusively within the CNS. Now through these interneurons, these motor fibers are sending inhibitory impulses to the sensory pathway. One of them is sending an inhibitory impulse to this presynaptic neuron, while the other pathway is sending an inhibitory impulse to the postsynaptic neuron. So in the sensory pathway, and we'll talk about that in another segment, but in sensory pathway, there are more than one neurons taking the sense to the higher brain centers. So you'd have, usually there are 3, sometimes more. So you'd have one neuron that would meet a second neuron. And again, where they meet is known as the synapse. Now these motor fibers are going to send inhibitory impulses either presynaptic or postsynaptic to the sensory pathway. So you have lots of different ways where the brain, or how we interpret and localize pain, or localize different stimuli. Now that's known as the central control of afferent information. So when we talked here about this lateral inhibition, this is pretty much happening at the levels of these primary neurons, or these first neurons, okay? While these are, oops, I'm going the wrong way, hold on. While these are happening at a higher level, okay, so this is what the CNS is doing in order to, so it's known as the central control of afferent information, and that is known as modification. Okay, so modification can either come from collaterals, like that lateral inhibition that we talked about, or these motor pathways, these descending pathways can send inhibitory impulses either to that first neuron, that would be a presynaptic inhibition, or to the second neuron, and that would be postsynaptic inhibition. Okay. So at this point, we'll stop, and we'll see you guys in part 2.